Welcome to the Sand Science Experience, your source for the most current and impactful research and preprints in science. Before we dive into today's fascinating topic, please take just a moment to hit that like button and subscribe to our channel. It truly helps support the Sand Science Experience and allows us to keep bringing you these in-depth explorations. Today we're embarking on a journey deep inside your very own cells. We're talking about mitochondria. Yeah, you probably know them as the powerhouses, but we're going way beyond just energy production. We're exploring a really crucial process called mitochondrial biogenesis. Sounds a bit technical, I know, but we're going to unpack it, make it clear, engaging, and hopefully reveal some, pretty surprising insights about life, health, and disease.
That's right. And, when we talk about mitochondria these days, we're learning they are just so much more than simple energy factories. Over the last decade or so, the evidence really shows they form this highly dynamic, interconnected network within our cells. Think of it less like individual batteries and maybe more like a constantly shifting power grid, always adapting. They're busy doing things like fusion joining together and fission splitting apart, and even cleaning house through something called mitophagy.
Self-cleaning. Wow. So it's not just about cranking out power. It sounds like it's tied into the cell's whole survival plan.
Exactly. I mean, beyond making ATP, which is the cell's energy money, these organelles, they regulate signals, they produce important reactive oxygen species. They handle fatty acid metabolism, they play roles in calcium stuff, amino acids, and even, the big decisions. Whether a cell lives, gets old, or dies is just a much bigger job description than we used to think. And key to all of this, for a healthy cell, is keeping up what's called mitochondrial homeostasis.
Homeostasis, that balance.
Yeah.
Right? We hear that a lot in biology. How does it fit here?
Precisely. It's this really tightly controlled balance. On one side, you've got generating new healthy mitochondria. That's our focus today, mitochondrial biogenesis. And on the other side, you need to efficiently get rid of the damaged ones, which is mitophagy. This constant cycle, this renewal and quality check, it ensures your cells can meet their energy needs, work right, and crucially, protect their own mitochondrial DNA. So our mission for this deep dive is really to get a handle on this intricate dance of mitochondrial biogenesis and understand why it's just so fundamental for health and for disease.
Okay, let's dig in then. We've heard the term. What is mitochondrial biogenesis really at its core? How does this self-renewal thing actually happen inside your cells?
Okay, simply put, it's the process where your cells make new mitochondria, but they make them from the ones that are already there. So imagine your cells need more energy, maybe after exercise, like you said, or if they're under some stress. Biogenesis is how they adapt. They launch this sophisticated program to make sure they have a healthy population of these organelles, the right amount, the right function, to meet whatever the demand is.
Okay, what's really wild to me is how do these things, which have their own DNA and are kind of semi-autonomous, actually get built inside the bigger cell? It seems like it must be a complex partnership.
That's a great way to put it. It absolutely is a complex partnership, and it goes back to their really unique history. Mitochondria were likely once independent bacteria, billions of years ago, absorbed by our ancestor cells, an endosymbiotic event. That's why they're semi-autonomous. They do make a few of their own proteins using their own mitochondrial DNA, the mtDNA, but, and this is the kicker, the vast majority, like over 95% of their proteins, are actually coded for by the cell's main DNA, the nuclear DNA. So those proteins get made outside the mitochondria, in the cytosol, and then they have to be carefully imported.
Wow. So it's literally a team effort between two different sets of instructions, two genomes. That's kind of mind-blowing. How on earth is that coordinated? It must need incredible precision.
It is incredibly precise. Yeah, it's this multi-step process needing exquisite coordination. So several key things happen. First, you need to turn on the copying of mitochondrial DNA. That's mtDNA transcription. This part is largely run by a protein called PGC1 alpha. You can think of PGC1 alpha as the master switch, or maybe the main conductor for the whole biogenesis show. When it gets activated, maybe through phosphorylation or other signals, it kicks off a whole cascade of other factors in the nucleus. And these, in turn, boost a key protein called TFAM. TFAM is like the foreman on the ground, directly handling the mtDNA transcription and replication. Okay, so that's happening. At the same time, the few proteins made from the mtDNA itself are being translated right there inside the mitochondrion. And also at the same time, that huge batch of nuclear-encoded proteins are being made out in the main cell area. These pre-proteins, they have like a special address label, a signal sequence that directs them straight to the mitochondria. Once they arrive, they get imported through these sophisticated tunnels like the TIM23 complex for proteins going to the inner membrane. And this import process, it's not free. It takes energy, both from the electrical charge across the mitochondrial membrane and from ATP. It's really a marvel of logistics.
Okay, this is where it gets really interesting for me. What does this all do for the cell? What are the payoffs? And how do scientists even track this? How do we know if biogenesis is happening well?
Right. Functionally, good mitochondrial biogenesis significantly boosts the cell's capacity for making energy efficiently through oxidative phosphorylation, fueling everything. It also helps cut down on harmful oxidative stress, which is key for preventing damage, and it can actively help repair mitochondria that are already having problems. As for measuring it, well, we look at a few key things. We measure how much new mitochondrial DNA is being made. We look at the levels of mitochondrial membrane components, like certain phospholipids, and we often look at the ratio, the mitochondrial DNA copy number compared to the nuclear DNA. An increase usually signals a growing, healthy mitochondrial pool.
That makes sense. But are there any catches? It sounds like it's always a good thing, but is it? Or can it go wrong sometimes?
Yeah, that's a sharp question. And yes, there are nuances. It's crucial to remember that just seeing more mitochondria doesn't automatically mean successful biogenesis happened. Other things affect the numbers. Also, while we often use TFAM levels as a marker, it doesn't always perfectly line up with the actual mtDNA copy number or the levels of all the mitochondrial proteins. And here's a surprising twist. In some very specific, unusual situations, biogenesis might actually cause problems, like maybe accidentally importing proteins that are misfolded, or even, weirdly, shutting down certain stress responses elsewhere in the cell. So it's not quite as simple as more is always better.
Okay, so if it's this vital, complex process, what actually turns it on and what shuts it down? What signals are telling the cell make more mitochondria or hold off?
Yeah, that's a huge area of research. Biogenesis is really tightly controlled by different signaling pathways that activate those key transcription factors, especially PGC1 alpha. And what's fascinating is how many natural things seem to induce it, like 6-gingerol from ginger or ursolic acid found in various plants. Interestingly, there aren't actually that many synthetic drugs specifically designed to just flip the switch on.
Can you give us a feel for some of the main signaling pathways involved, the big on switches?
Sure. One of the most studied is the AMPK PGC1 alpha pathway. AMPK is like the cell's fuel gauge. When energy runs low, think exercise or cutting calories, AMPK gets activated, and that boosts PGC1 alpha, which triggers biogenesis. AMPK also helps loosen up DNA packaging, making genes easier to read.
So that's part of why exercise is so good for mitochondrial health.
Exactly. Then there's also the direct induction of PGC1 alpha along with its buddies NRF1 and TFAM. We see this with certain treatments like in pancreatic cells or even with salicylate, which is related to aspirin. There's also the CRB-PGC1 alpha pathway, often triggered by receptors on the cell surface. A type 2 diabetes drug, lixisenatide, actually uses this route. And then you have others like the G-beta gamma ACT pathway, important for recovery processes, and even dietary stuff influencing the ACT-FOXO3A axis. And we can't forget the sirtuins. SIRT1 works with PGC1 alpha, and SIRT boosts proteins involved in energy production and fat burning. It just shows how interconnected it all is.
Wow, that's quite a network of controls. It really highlights how central this process is. Okay, what about the flip side? What turns it off? What inhibits biogenesis?
Right. The inhibitors generally work by suppressing those key factors like PGC1 alpha, TFAM, or AMPK. Examples include things like a specific microRNA, miR130BP, some synthetic compounds like one called XCT790, and perhaps more relevantly day-to-day, a persistent environment of high glucose and high fat.
Like in metabolic syndrome.
Exactly. That kind of environment can dampen biogenesis. Also, some local anesthetics, certain types of nanoparticles, even a hormone called resistin can put the brakes on it.
And what happens inside the cell when this vital renewal process gets slowed down significantly? What's the downstream effect?
Well, reduced biogenesis often goes hand in hand with other mitochondrial problems. The cell's ability to make ATP efficiently can be impaired, and maybe counterintuitively, it can sometimes speed up mitophagy, the removal process, maybe as the cell tries to clear out a higher proportion of damaged mitochondria when it can't make enough new healthy ones. You also often see that ratio we talked about, the mtDNA to nuclear DNA ratio, go down, suggesting a less robust mitochondrial population.
Okay, so we've covered the how and the what influences it. Let's get to the really crucial part, the so what. Why should you, listening right now, really care about this intricate cellular mechanism? What does it mean for your health?
This is incredibly important because when mitochondrial biogenesis goes wrong, either too little or too much, it's linked to a whole host of diseases, and that means it's a potential target for new treatments. We basically see two main scenarios where this matters hugely for health. Situations where biogenesis is impaired and we need to boost it.
Right.
And perhaps surprisingly, situations where it might be abnormally intense and we might need to dampen it.
That's right.
So first, let's talk about impaired biogenesis. Where do we see this? A big one is aging. As our cells age, as they become senescent, they often show reduced mitochondrial biogenesis, AMPK activity goes down, levels of PGC1 alpha, TFAM, SIRT1 drop off. So strategies to slow this down involve things that stimulate PGC1 alpha compounds like resveratrol are studied for this, but also things like regular exercise and calorie restriction, basically keeping your cellular power grid tuned up as you get older.
So that run isn't just good for your heart. It's literally helping maintain your cells' energy factories over time. That's pretty motivating.
Absolutely. Then you have metabolic diseases like type 2 diabetes and obesity. Again, impaired biogenesis is often part of the picture, linked to that sluggish PGC1 alpha AMPK signaling. Endurance exercise is a standard recommendation here, partly because it boosts biogenesis. Researchers are also looking at things like spirulina or certain plant compounds to help promote it, especially in fat cells and obesity.
Okay, aging, metabolic disease. What else?
Neurodegenerative diseases. Think Alzheimer's, Parkinson's, Huntington's, ALS. Impaired biogenesis is a common thread in many of these. So therapeutic ideas often revolve around finding ways to augment it, maybe using compounds like melatonin or berberine. What's really interesting here is that sometimes an early response to the stress in these diseases is actually an increase in mitochondrial DNA replication, almost like the cell is trying to gear up for biogenesis as a defense mechanism.
So for something like Alzheimer's, the hope is maybe helping those neurons generate more healthy mitochondria to better withstand the disease process.
Exactly. Bolstering their energy supply and resilience. Now the other side, abnormal intensification. Where might we need to turn it down? This is often seen in some types of aggressive cancer. Certain cancers seem to rely heavily on enhanced energy production through mitochondria, and increased biogenesis correlates with their ability to invade tissues and metastasize.
So the cancer cells hijack the process for their own growth.
In some cases, yes. So strategies there might involve inhibiting biogenesis, using specific drugs, or maybe even repurposing things like the antibiotic doxycycline, which seems to hit cancer stem cells partly by affecting their mitochondria. But it's complex. Some standard chemotherapy drugs can actually paradoxically stimulate PGC1 alpha. So it really depends on the specific cancer's metabolic wiring.
It's definitely clear that whether it needs turning up or dialing down, mitochondrial biogenesis is right in the thick of it for cellular health and disease. It's not just some obscure pathway, it's fundamental. So what's the main takeaway for our listener from all this?
Yeah, the big picture is that mitochondrial biogenesis is this incredibly complex but intensely studied biological process. It's how these essential organelles renew themselves, maintain their DNA, and ultimately ensure the whole cell stays in balance in homeostasis. And PGC1 alpha really stands out as that master regulator, that central hub, responding to all these different signals, activators, and inhibitors. Its role is just critical.
And looking forward, what are the implications for medicine for health? Where is this research heading?
Well, the fact that messing with mitochondrial biogenesis pops up in so many conditions, aging, diabetes, neurodegeneration, cancer, just screams therapeutic potential. We've made huge strides in understanding the how, but there's still plenty of questions like, are there unique mechanisms in neurons? What's its exact role in heart disease or arthritis? But the prospect of developing new therapies that specifically target mitochondria, maybe to boost biogenesis in degenerative diseases or carefully inhibit it in certain cancers, it's really exciting. It could lead to totally new ways to treat chronic diseases, maybe even prevent some aspects of aging or cancer. It really makes you wonder, doesn't it? How might learning to precisely control your own cellular power plants change medicine in the future? What could that mean for health and longevity?
Definitely food for thought. We hope this deep dive into mitochondrial biogenesis gave you some powerful new insights into the amazing world inside your cells. If you found this information valuable, please do hit that like button and subscribe to the Sand Science Experience for more deep dives into current and impactful research. Your support really does make a difference. Thank you for joining us on the Sand Science Experience.